Good afternoon everybody. Today I'm going to be talking about cellular metabolism, cell growth and division in cervical cancer analysis. This presentation provides an in-depth analysis of cervical cancer, focusing on principles of cellular metabolism and cell growth and the divisions of mechanisms. Protein synthesis begins with DNA unwinding, allowing transcription of genetic codes into mRNA. This mRNA is then translated into proteins by ribosomes, the tRNA playing a crucial role in amino acids. So DNA packages round the proteins called histones. Enzyme polymerase attaches to the start of the gene strand, and the process of adding and removing DNA strands begins. This process is called transcription. Once the mRNA is formed, it moves out of the nucleus into the cytoplasm towards the ribosomes. The ribosomes reads codes within the mRNA and produce a chain of amino acids. The amino acids are molecules that combine and form proteins. They're the amino acids. Each amino acid usually matches with another amino acid. So, for example, thymine goes with adenine and guanine goes with cysteine. The tRNA carries the amino acid into the ribosome, and the mRNA is read 3 bases at a time. The tRNA, T stands for transfer, delivers the corresponding amino acid as each triplet is read. The chain then folds into a complex protein once the final amino acid is added. Next I'm going to be talking about DNA replication stages. So initiation. Initiation proteins attached to certain sites in their replication origin are forming a complex DNA and proteins. Elongation, DNA synthesis takes place on the 2 strands that make up the double helix in opposed directions. The leading strand is created in the short segments called Okazaki fragments. RNA primers are inserted into the exposed bases of the lagging strand, and DNA synthesis then continues in the 5' to 3' direction. Termination. Termination is 2 replication forks meeting at the same DNA. Same DNA strand. Determines DNA replication at this point. Replication proteins, kinetin, and any gaps are filled, ligated, while all intervening DNA is unravelled. Next topic is stem cells. There are 2 types of embryonic stem cells, totipotent and pluripotent. Adult are multipotent and oligopotent and unipotent. Embryonic stem cells, known for their pluripotency, can differentiate into any cell type. This contrasts with multipotent adult stem cells, The formation of specialised tissues from these stem cells is a complex process influenced by genetic and environmental factors.
Compliments.
Next topic is going to be interphase and cell division. Interphase is a critical phase of the cell cycle, involves cell growth and DNA replication, setting the stage for accurate genetic material divisions in daughter cells. During the interphase stage of the process, that's where the cell usually grows and gets stronger. Next subject is initiating cell division. Cell division is regulated by checkpoints and proteins that ensure optimal conditions for division, maintaining cellular integrity and function. There are 4 checkpoints in a 24-hour period. That's a 24-hour period. Starting at G1, at this stage cells such as G1 is the longest phase, where the cell begins to grow. At this stage, cells such as neurons, which are fully developed, neurons of the brain, are able to exit the cycle. G2 is the shortest part of the cycle. This is the final stage before mitosis. Genetic information in daughter cells. Mitosis ensures identical chromosomes distribution to daughter cells, while meiosis introduces genetic diversity essential for evolution. The important part of this is each cell has to be identical to be able to create mitosis. Meiosis is a process of cell division that creates gametes for sexual reproduction. This is similar to mitosis in its 4 phases, but happens twice. In meiosis, spindle fibers pull apart chromatids, which are replicated chromosomes composed of identical halves joined at the centromere. So as you can see here, the process is being shown on the diagram. The chromosomes start to pair up during and go down the centre of the cell. Chromosomes swap sections of the DNA. And this creates a mix of new genetic material in an offspring cell. Spindle fibers then start to take the chromosomes to the opposite poles of the cell. The nucleus divides twice into the daughter nuclei. The daughter nuclei have single chromosomes, a new mix of genetic material. Next subject is mutations of chromosomes. Mistakes during cell division. So mitosis is the process of duplicating cells. The main purpose is for growth and repair. So I need to talk through these phases with you, sorry about that. So you've got the interphase, which is the resting phase where it grows. And then you've got the prophase, which chromosomes are visible and the nucleus is still intact. The metaphase, nucleus is being disassembled and replaced with chromosomes. The anaphase, chromosomes are separated by spindle fibres and pulled towards the poles of the cell. In telophase, chromosomes move to opposite ends and form new nuclei, resulting in two new cells. Cytokinesis splits the cytoplasm to a complete mitosis. Mistakes during cell division can cause chromosomal mutations, leading to breakage, duplication, or movement of segments between chromosomes, impacting whole genes and chromosomes instead of specific nucleotides. This is where genetic deformities can happen and mutations such as cancer. Which brings me to my next subject, which is my chosen cancer, which is cervical cancer. Cervical cancer cells exhibit uncontrolled growth and can invade other tissues unlike normal cells. These changes result from mutations in key regulatory genes. Normal cells typically grow at a regulated standard rate. They grow, divide and die, whereas cancer cells grow recklessly. Instead of dying, they keep dividing and growing too rapidly to fully develop. Cancer cells are invasive, influences normal cells and affects the immune system. They can easily spread to other parts of the body, in this case vagina, lymph nodes, bladder, intestines, lungs, bones and liver. Cervical cancer is a type of cancer that occurs in cells of the cervix. It is characterised by abnormal growth and can spread to other parts of the body. Treatment options for cervical cancer include surgery, radiotherapy and chemotherapy. Research is ongoing to develop vaccines and targeted therapies to improve treatment outcomes. So during my research I've developed a diagram which is the breakdown of my chosen cancer. So the characteristics of the cancer cell are morphological changes, irregular cell shape, and enlarged nucleus. Genetic alterations, which HPV integration, mutation in tumour suppressor genes, and chromosomal instability. Metabolic changes are increased glucose uptake, altered lipid metabolism, and enhanced amino acid metabolism. And invasive behaviour is loss of cell adhesion, increased motility. The risk factors are the virus is usually created by an infection called HPV. HPV virus is widely spread, and currently they're doing vaccines in schools to make sure that this sexually transmitted infection doesn't keep growing, and therefore hopefully reducing the risk of cervical cancer in the future. Smoking is another risk factor to contract the cancer, and long-term use of oral contraceptions. Symptoms include abnormal vaginal bleeding, pelvic pain, pain during intercourse, and struggling to go to the toilet. The diagnosis is done by a pap smear, HPV DNA test, and a biopsy. And the treatments are surgery, radiation therapy, and chemotherapy. This presentation covers cellular metabolism, growth division, and cervical cancer analysis, stressing the importance of ongoing research and treatment advancements.